Okay, good afternoon, everybody. Today I'm here to talk to you guys about a really cool and unique form of twitching or motility called twitching motility in a disease-causing bacterium called Pseudomonas aeruginosa. And I'm going to discuss how this is regulated by a system of navigational proteins. The first panel here shows a representative image of twitching cells. What you can see is that they move in one big group and in one specific direction. If you zoom in at the leading pole of one of these cells, as I've done in the center panel, you'll notice these really thin hair-like fibers, and these are called type 4 pili. They're what propel a cell during twitching motility. They work by going through repeated cycles of extension and retraction to pull a cell along a surface. Essentially, they work a lot like Batman's grappling hook. Now, if you zoom in further to the molecular level, and the schematic on the right, you'll notice something similar to this at the pole of the cell. The shapes in red represent the proteins that make up a type 4 pilus. Of particular importance to my project are the three different motor proteins, which I've labeled here as B, which builds up or extends a pilus, T, which retracts or tears down a pilus, and U, whose function is unknown. The shapes in blue represent the navigational proteins I alluded to earlier. Their job is to sense the chemical environment around a cell, and in response to specific chemicals, they'll regulate pilus function and make the cell move either towards or away from these chemicals. Now, I studied two different aspects of the signaling pathway. The first one is what happens between the chemical sensing step and between the type 4 pilus functioning step. A number of proteins have been identified by our group, as well as other groups, that fall within this intermediate zone. And what I'm trying to do with my research is determine whether or not these proteins regulate pilus extension or pilus retraction. The other aspect that I look at is the function of that third motor protein, U. Now, its function has really been a mystery in the pilus research field for quite some time. However, what we've observed is that U is the only one of the type 4 pilus proteins that's found at one pole of the cell. All the other ones are found at both poles of the cell. So what we hypothesize is that this motor protein serves as a direct link between the type 4 pilus and the navigational system. That being said, what we believe is that signaling through the navigational system will make U go to one end of the cell, and it would mark that end as being the front, thereby imparting directionality on twitching motility. So in the end, why is this process so important to study? It's been shown by many groups that twitching motility is a really critical process in infection with this bacterium. So as the saying goes, knowledge is power. So having a better understanding of how the pilus works and how it's regulated will hopefully reveal potential targets that can be exploited in the future for therapeutic use. Thank you all for your attention.